viewers, welcome to CEC Gurukul lectures, and I am Dr. Sanavar Soham from Department of Botany, Kalindi College, University of Delhi, and I have been discussing plant systematics. Now in this lecture, I will be discussing some other important aspects of plant systematics, and I will be beginning with phonetics and cladistics. Now, the key difference between phonetics and cladistics is the method used in classifying organisms. Phonetics classifies organisms based on morphological and structural features, while cladistic classifies organisms based on their ancestry and evolutionary relationships. Classification of organism is an important process in biodiversity and biological studies. In this regard, phonetics and cladistics play an important role in building relationships among organisms. Now, what is phonetics to begin with? Phonetics is the area of study that classifies organisms based on their similarity. It may include classifying organisms based on their structure, based on morphology, and various other traits that are observable. Phonetics disregards evolutionary relationships. It is thus a primitive way of classifying organisms. However, phonetics includes the usage of various techniques in order to build relationships among organisms. Clustering and ordination are two ways in which organisms undergo phonetic classification. During phonetic classification, clustering or ordination of organisms take place based on phenotypes that could be observed by the taxonomist or the scientist. Therefore, dozens of characters are analyzed before clustering the organisms. Then, the representation of these characters using graphs takes place. This then leads to grouping of the organisms. The main drawback of phonetics is that there can be misinterpretation of organisms into groups as only physical visible characters are considered. This could give false results during the classical clustering process. Therefore, in classifying, it is important to analyze many methods or grouping for accuracy, especially the phylogenetic data of organism. Now moving on to cladistics. It is the study area that classifies organisms based on their ancestry. Thus, cladistic considers evolutionary relationships. In cladistics, the most common ancestor, which is the recent one of a particular organism, plays a key role in classification of that organism. Therefore, cladistic depends on evolutionary relationship of organisms, since the similarity between organisms relies on the evolutionary patterns. In generating a classification of organisms based on cladistics, a cladogram is used. A cladogram is a tree-shaped diagram. Initially, during the generation of cladograms, physical and morphological characters were used. However, at present, cladistic mainly depends on genetic sequences and phylogeny. Therefore, cladograms are also referred to as phylogenetic trees sometimes. Furthermore, at present, cladistic uses morphological, evolutionary, and phylogenetic data in classification. The branches of the cladograms define the various distances of similarity between the organisms and the deviation of the organisms. The accuracy of classifying organisms is based on cladistic is more accurate and efficient because it is letting you know about the similarity of the organism and from where and which point it deviated during evolution. Bioinformatics play an important role in cladistics, increasing the accuracy and acceptability of the results. Now this diagram shows the phylogenetic tree, or also known as cladogram. As I told, there are branches known as clades, and the deviations of the branches show from which point it basically diverted from the main clade or the branch. Now we will move on to another important aspect, which is known as the species concept. The taxonomists are primarily concerned with comparisons of species rather than individuals or parts of individuals. Therefore, it is very essential to define and delimit a species first. The question of what is species and how speciation occurs have accumulated voluminous literature, but with different conclusions. In the beginning, species were supposed to be created as separate entities, as static and monotypic. There was no serious species problem. But today, this concept is no longer valid, and there has been a greater emphasis on the dynamic concept of the species. Various attempts have been made to define a species. So, broadly 5 different species concepts are provided by the scientist. These are, the first one being nominalistic species concept, which believes that nature produces individuals and nothing more. Another one is the typological species concept or taxonomic species concept, a very natural group of organisms. Hence, a natural taxon in classification has an invariant, generalized or idealized pattern shared by all members of the group. The third one is the biological species concept, which was given by Dobzhansky in 1937, and it is an important species concept. Groups of interbreeding naturally populations that are reproductively isolated from such other groups have been defined as biological species by Dobzhansky. Then another one is alternative species concept or evolutionary species concept, according to which a spatio-temporal lineage of population that evolved separately from other lineages have been termed as a species, and it has its own ecological niche. And the last one being ecological species concept, which was given by Van Wellen in 1976. According to this, the species is a group of individuals which is maintained ecologically but not reproductively. Classical taxonomists define species as mainly a morphological category intended for herbarium classifiers and having certain common morphological characters. This is known as morphological species concept, which is based generally on the morphological characteristics. Biosystematics defines species in terms of breeding populations, common gene pools, reproductive isolation, as well as breeding behavior. This is known as biological species concept, which was first introduced by Dobzhansky. The biological species concept defines a species as a group of interbreeding populations reproductively isolated from one another, such group of populations. According to this concept, species are classified by only one criterion, as I told you, which is reproductive isolation. So, the biological species concept has well served the purpose of animal taxonomy, but in case of plants, it has created one confusion. Unlike animals, many plants hybridize freely in nature, as we very well know, and are thus not reproductively isolated. In the genus Epilobium, which has about 160 species which are interspecific, and they undergo interspecific hybridization, is very common. According to biological species concept, the whole group is to be placed under a complex species. There are very many examples of this kind. In the Orchidaceae and Poaceae, distinct genera freely hybridize in wild, and the amalgamation following biological species concept would result in large heterogeneous genera, which is of little practical value. Some such cases, for example, Festuca, Lolium, and Vulpia complexes in Poaceae. Conversely, there are species such as Capsula bursa pastores, Vicia sativa, and Senecio vulgaris, which include taxa morphologically indistinguishable, yet they are incapable of interbreeding. Although it has now been known and shown that in too many instances this concept is inapplicable to plants, the enunciation of biological species concept was great advancement in the history of systematics, without any doubt. Any consideration of solution to species problem must take into account the current views, such as all populations tend to vary and that no two populations are ever alike. Second, that some of these variations are adaptive and are of survival value. Then, the forces of nature result in extinction of some individuals while others survive with the same force, and that some of the variations shown by individuals within a population must be hereditary if successive generations are to be modified from the ancestral types. And the last one, environment of the individuals must not be static. These views collectively incorporate the principles of the theories of natural selection and evolution of species. With the introduction of biological species concept in plant systematics, morphology is considered by experimental taxonomists to be a weak criterion for species delimitation. Ayala have even remarked that morphological changes is neither necessary nor sufficient for speciation to occur. However, as taxonomy is concerned with variation patterns and their classification, an evolutionary synthesis that does not explain morphological change is at best incomplete. Another critical remark made by some on biological species concept is that this biological species concept has its main roots in population biology, which involves fertility tests, and such tests have been restricted to few selected individuals of a population, and the results have been generalized to apply to all populations and species. Added to these, there are reports of many natural, interspecific, and even intergeneric crosses just mentioned above, which have posed great problems to evolutionary biologists. Therefore, Sokal and Crevallo concluded that biological species concept is neither operational nor heuristic nor of practical value. They feel that phonetic species concept is more appropriate one to be associated with the taxonomic category species. Early taxonomists did not know that individual species possessed certain degree of capacity for variation, so they thought that the description of a single plant would furnish the description of species to which it belonged, and thus came the population concept. But today, all taxonomists would agree that this method is inadequate and that they should study the variations that exist in one and the same species. In other words, they should study the group which individual forms, that is, population. The term population thus refers to those group of plants that grow in a particular habitat at a particular time. Several populations may constitute the species. In such cases, there are bound to be discontinuities between different parts of the population. The more widespread such population systems are, the more likely there are to be appreciable differences between them. A local breeding population whose numbers grow together and can interbreed to form a common gene pool is recognized by the biosystematists as the unit of evolutionary change. Such panmictic populations between whose members there is potentially a free gene flow shows variation in many features even though they belong to the same species. Populations are therefore dynamic and show variations within populations and variations within the populations of the same species. Variations between individuals is caused by factors such as modifications due to the external environment, or maybe mutation, or even genetic recombination. Now moving on to the role of ecology in understanding of species. It is now clear that phenotype is not merely a manifestation of the genotype, but the environmental factors play an important part in modifying the latter to produce the former. There are many instances in our local flora where the same genotype can produce a range of phenotypes through development in different environment. Such variations are termed phenotypic modifications or ECADs or ecophenes, and this phenomenon is known as phenotypic plasticity. Environmental factors may include factors such as light, temperature, wind, crowding, soil, altitude, etc. and they form a complex in determining the phenotypes of populations. More often, it can also be observed that different populations of a species occupying distinctive habitats may show different structural and physiological features, often of an adaptive value. The classical experiments of Turisson on gene ecology have clearly demonstrated this phenomenon. He called these genetically adapted ecological races as ecotypes, which can be equated with the varieties of classical taxonomies. Several such ecotypes together form an eco-species, which can be equated to species of the morphologist. He also gave the species concept, which was mainly based on ecological factors and was widely used by experimental taxonomists. On the other hand, Van Valen, supporting the ideas of Turisson, suggested that species are maintained for most part ecologically and not reproductively. He proposed the ecological species concept, and the basic assumptions are: number one, that the genes are of minor importance in evolution and should hence be considered on par with other molecules. Secondly, the control of evolution is largely by ecology and the constraints of individual development as well. Third, the selection acts primarily on the phenotypes, which are the building blocks of a community, and not the genotypes. So, we move further with the concept of speciation. It is the process by which new species are formed from the ancestral ones. The most widely accepted hypothesis explaining the process of speciation is the geographical theory of speciation, which is also known as allopatric theory of speciation. According to this theory, the first step in speciation is reproductive isolation, which is brought about by the physical separation or geographical separation of the populations. The separation results commonly from a shrinkage of the original range of a species so that the populations become isolated physically. The separation may also be due to the result of large range of dispersal when new populations become established beyond the range of pollination of the original gene pool. A second step is the independent differentiation and evolution of these reproductively isolated populations. Eventually, if the ranges of populations merge again and the reproductive isolation persists, then the speciation process is considered complete. Ecotype formation just discussed above is the first step in speciation, in differentiation. Several examples of ecotypic differentiation are available. For example, Agrostis tenuis, a temperate grass, also grows on lead concentrated soil and has become lead tolerant. This character got fixed and not lost even in the absence of the metal. But still, the two populations growing side by side may cross and produce hybrids that cannot tolerate the lead concentration on one side or cannot compete with the other normal plants of the species on the other side. Thus, the lack of hybrid survival constitutes absolute barrier to the gene exchange. Sometimes, speciation may take place abruptly, also by the development of polyploids and mutations. According to Futuyama, speciation is more thoroughly awash in unfolded and often contradictory speculation than any other single topic in evolutionary theory. Now, there are different isolating mechanisms which operate to prevent or reduce the gene flow between the populations. Spatial or environmental isolation prevents gene exchange between populations because of the distance between them. Reproductive isolation depends on some aspects of plants themselves, such as lock and key pollination mechanisms. Difference in flowering times of two populations, etc., etc. Salvia apiana and Salvia mellifera, for example, are two species that grow in different zones, but at some point their ranges overlap. Salvia apiana shows a preference to the drier soils, although the two species often are found intermixed, whereas Salvia mellifera blooms in April, the other one blooms in May and June. So consequently, there is a little overlap in their flowering times. Salvia mellifera has smaller flowers which are pollinated by smaller bees, flies, butterflies, etc., while other species have much larger flowers and are pollinated by larger carpenter bees. These ecological and mechanical differences combine to keep the two species reproductively isolated. Nevertheless, hybrids are formed occasionally, but hybrids are only partially fertile. This illustrates how species with no absolute genetic isolation barriers can grow side by side with no hybridization. Now, moving on to taxonomic hierarchy. To begin with, each and every plant organism has to be provided a scientific name, which is a binomial comprising of a genetic name and a specific epithet. Such species are then grouped together into a higher taxon, a group or category called the genus. The genera are then grouped into a still higher unit or taxon called the family, and so on. In this manner, it is possible to reach the highest level in system of classification. This ultimate unit or taxon is the plant kingdom or even the domain. The sequential placement of more than one taxa, one above the other or one inclusive of the other, is known as taxonomic hierarchy. Generally, the taxa are arranged in ascending order, the highest one being the kingdom and the lowest one being the species. All of these categories, ranks taken together, constitute taxonomic groups. Mayer introduced the term taxon for the first time, and it was finally adopted in an International Botanical Congress which was held at Stockholm. Taxon is thus regarded as an adequate, appropriate and specific term to designate an entity or a taxonomic group. The word entity is not preferred because it cannot designate a biological category or a taxonomical group above the level of an individual. The term taxon has many advantages. I will quickly tell and discuss them. It possesses a single meaning that is devoid of any vagueness or ambiguity. It is descriptive. It is used to indicate any of the taxonomic groups of any rank. These ranks could be division, class, order, family, genus, or even a species. For example, division Magnoliophyta is an example of a taxon. So are the orders Rosales, family Asteraceae, genus Mangifera, or species Ilium cepa. They all are known as taxa. Now I will close this lecture with the taxonomic hierarchy, and I will discuss various aspects of it in the upcoming lecture. Thank you.